So
Okay, now we're live. All right. Hello, everybody. This is Haley and Katie. So our partner, Lauren, we had some scheduling conflicts. She's going to be doing a video all by herself, and we're going to be doing a video together because it is the last video, and we understand that the conflicts are annoying, but they just happen. All right. And so Haley and I are going to be doing 1 through 5. I'm going to do questions 1, 2, and 5. And Haley is going to do 3 and 4. All right. So I'll start off with number 1. Number 1 is what would the banding pattern look like on a gel if we had this sequence of DNA? And I have it drawn out here. So you would have your G's. So you'd have about 3 bands on your G line. You have about 4 on your A line, 3 on your T, and 3 on your C. And then for for B, we have the DNA polymerase would denature due to the extreme heat that the TAQ can handle. And then for C, I put it would interfere with the DNA primer and prevent binding. And then for D, it would only the bands on the DTAP would show up. So basically, you'd only see this. You wouldn't be able to see any of these other ones because it doesn't have what is needed to show, oh, this is where the G is, this is where the T is, this is where the C is. And that is number 1. Number 2 is Taq polymerase synthesis. So during a typical PRC cycle, the Taq polymerase synthesis steps is done at 70 degrees Celsius, while the primer anneals to the complementary site is done at 55. Then we have NPR is the common selectable marker that is found in many plasmid vectors, while the restriction endonuclease is recognized in cleaved DNA molecules at the palindromic, I'm sorry, I'm going to botch like almost all these words, palindromic site. C is the ergos gel electrophoresis. DNA is placed on the electrical field. DNA migrates towards the positive electrode during the electrophoresis. D is DDATP lacks the 2OH group and the 3' OH group. The competent is the term, competent is the term used to describe the process where where bacteria takes up DNA molecules from the environment. And F is during a blue-white screening, IPTG is commonly used to induce the expression of the lac operon, while X gal is cleaved by the beta-galactyl, I'm not even going to try, to turn E. coli cells blue. All right. And Haley, you are a go.
awesome. Okay, so for 3, let me get back to you guys. For 3, it says you were interested in using a PCR to amplify regions of a double-stranded DNA fragment. That is to be copied is located between sequences in bold below. So this is the sequence to be copied. It was really hard for me to see the bolded parts, so I highlighted them. So write a single-stranded DNA sequence that you would use for the 2 primers. So I wrote 2. I wrote both of them here and here because I didn't know exactly if there was one specific she wanted. So you just take the primers off and use the DNA in between. And then the mathematical equation, the temperature, so right here she gave it to us. The percent GC is the primer expressed in integers, not as a decimal. So I'll explain what that means a little bit later. And the N is the number of nucleotides within the primer. So for the first one, I think it's this one, but I'm not sure. So this is your primer area. And so you count how many nucleotides are in the primer. So I think there's 16. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. And so that's your N. That's the number of nucleotides within the primer. And then you go and count all of the G and C nucleotides. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is the first primer right here. And so when the GC nucleotide primer, so what I did is 9 divided by 16, and that's how I got my 56.24% GC. And then you plug that all into your calculator, and you get 39.54 degrees Celsius for that the green primer there. And you go through that same process. There's 16 nucleotides again in the yellow primer, but there's only 8 of the GC, so therefore it's 50%. And then you plug it all into your calculator again, and you get 39.51 degrees Celsius for those temperatures. And then each PRC cycle contains a DNA synthesis step. What are the sequences of the two newly synthesized strands of DDCTP is accidentally included in the PCR experiment? Assume that DATP, DTTP, and DGTP and all other appropriate PRC components are found in the reaction mixture. So what this means is that once you get to a C, it will stop the PRC experiment. So for the first one, for 5' to 3', you come up here, and it'd be GG, so right here, GG, GATTAGC, and that's where it will stop because you hit that C. And then for the second one, as you go from 5 to 3', it goes TTT, AAT, and then you hit a C, so then it would stop there as well, right there. That's where we got those. And for D, it says, when we learn about DNA replication in E. coli, we learn that DNA A and DAM. Why are these two proteins not included in the PCR? So I put DNA A separates two strands of DNA, but because of the heat, it's not needed, because the heat denatures the DNA and it splits by itself. And the DAM is not necessary because the PCR experiment primers are used for starters, and so additional starters are not needed, not necessary. And then for 4, pretty beefy. 4, the question concerns Argos gel electrophoresis and restriction mapping. Suppose you set up 6 restriction enzyme digest reactions contain the same linear with different combinations of restriction enzymes. We have BAM-HI, ECO-RI, and HIND3. So this is what it produces. For A, it says draw an image that represents the result of the Argos gel. So she gave you the base pairs she wanted on there. So I have my 100, 200, 300, 400, 500, and 1,000. And so basically you're just going up here. So the BAM-HI, which is one right here, you go up to 50, draw a line, go up to 350, draw a line, 500 base pairs, draw a line. And so you do that for all of your DNA fragments, for all these right here, in that match up with these. So then it'll look something like this. Then for B, it says draw a linear restriction map that indicates the location of restriction enzyme sites along the hypothetical DNA molecule. So for B, it's using the same numbers as in that chart. So these numbers, but you're drawing it on a linear map. So you'll get something like this for B. So 50, 350, 500, and so on and so forth. But once you get to the fourth one, that's when it starts to get a little trickier. So for the fourth one, you're combining these two. They're both that way, and then you figure out which one cuts where. So the BAM cuts after 50. You can see here it cuts after 50. And up here. then the eco cuts there, and then the BAM cuts again. So you're kind of combining these two right here. And same with the BAM. That's supposed to be an H. I got sloppy. So the BAM and the what is it called? Hind 3. And then we'll do that for all of them. And then for C, what are the predicted fragment lengths of this linear DNA molecule that's cleaved with 3 molecules simultaneously? So the BAM always cuts at 50. So they're always right here, 50, 50, so it'll cut there. Between BAM and ECO, there's approximately 150 right here from the previous one. So that would be 150. And then between E and H, there's, okay, and then BAM cuts at 500, and then H and B is 100, and then what's left over is 200, so that would be in the middle. And that's how you do number 4. And that's C's pretty tricky, so I'd go watch her video, and she explains it a little better than I did. So.
Okay. Let me go back up there. All right. Okay, and then for number 5, it's asking us to do the steps if we were to take this yeast. I'm just going to call it SC. And we're trying to find this gene called YFG. And this is the process in which you would take it through in order to find that YFG gene. So you'd start by making cDNA, which you start with purified mRNA. You mix it with poly-DT primer, add reverse transcriptase and dNTP to synthesize the DNA strand. You add RNase H to digest the mRNA. By cloning it, you would add the DNA polymerase to make the second DNA strand. Then you make the plasmid vector, which is the plasmid is isolated, and then the plasmid is digested with a restriction enzyme, then separated by gel electrophoresis. And then the DNA fragments that you'll see are markers, so that way you can find the location of the restriction enzyme. And then you need to do blue-white screening, which is you add the cells after transformation to a media. That includes IPGT and ampicillin. And this induces the lac operon and the XGAL. So if the beta galactose something is converted into XGAL, then the colonies will be blue. And if the beta is not converted into XGAL, then the colonies will be white. So you'll know if it's present or not. And that's going to be it for our lesson 13 group problem. Thank you for watching. Okay.